Warahmatullahi wabarakatuh. Hello students, how are you today? Before we start the class, I want some of you to tell me about your condition. So, can we start from Rizka? Yes, please tell me your condition. Alhamdulillah, I'm very great that you're doing great today. Can we move to Miss Kiki? Tell me about your condition. Great, it's very great. So, me and Mrs. Mary today will teach you about Kingdom Monera. So before we start right into the lesson, please open page 43 in your book. Done? So can we move to this picture right here? Can you guys tell me what organisms they are and what kingdom they are from? Yes, correct. It is from Kingdom Monera. Specifically, they are bacteria. It has unique shape, right? Can you guys name what shape they are? Yes, it is bacillus. The bacillus term means that the bacteria shape is like stick. Do you understand? So, can you guys name some organisms, some bacteria where you can found in your daily life? Maybe you remember some of them? Yes, of course, we have E. coli, or as known as Escherichia coli, where we can find it in ground beef, unpasteurized milk, and fresh food where it hasn't been cooked yet. And they can cause symptoms like vomiting and diarrhea, where it can get worse if we don't treat them right. So let's move to the actual lesson. So today's actual lesson is about Monera, also we know them as bacteria. The term bacteria comes from the term bacterium, which means small rod. They are very, very small in size, and they are unicellular organisms, and they do not have nuclear membrane, which is why they called prokaryote organisms. And also, they were first found by Anthony van Leeuwenhoek in 1674, and usually they have cell walls. They are microscopic. Like I said before, they have very, very small size, so they have to be seen under microscope in order to be observed. They cell diameter usually are about 0.5 to 5 micrometers. Monera basically divided into two major groups, which are Archaebacteria and Eubacteria. And actually, from their names, we can give them some definitions. The word archae in Archaebacteria comes from Greek language, which means ancient or old. The member of this group has been living in this earth since Earth's early stage, and they live in extreme environment and believed as one of the earliest cells that live in this earth. And Eubacteria, the word eu means true. The member of this group are prokaryote organisms, and they can live literally everywhere in this earth, or as known as cosmopolite organisms. See bacteria cell structure next. So, basically, the bacteria cell structure is not as complex as other cells that exist in this world. They have pillars, they have capsule, they have plasma membrane, nucleoid or DNA as genetic material, and they have cytoplasm, ribosomes, and flagellum. So the capsule of the bacteria cell structure is the outer part of the cells, which helps them to keep the cell from dehydration, and they help the cells to attach to other bacteria or substrate. And then cell wall helps them to maintain cell shape, and then it is composed by peptidoglycan, or it could be not. And then plasma membrane is one of the universal composer of cells. Without them, the organelles inside the cells will be everywhere. So the plasma membrane consists of phospholipid and protein, which is selective permeable, and then it has role in substance exchange. And cytoplasm, as you know, it is basically are the place for every organelle in cells. such as ribosomes here and nucleoid or DNA. And cytoplasm is also a site for metabolic reaction. And we have ribosomes. Ribosomes is known as their function in protein synthesis. So we have pilus and flagellum here. Basically, they are composed by the same organelles, which are microtubules, but they have a difference in their function, which is the pilus is to attach to other bacteria or substrate, while flagellum is for movement. But this structure of flagellum here is not exist in every bacteria. It depends on the type of the bacteria. Organelles of the bacteria cell structure is this: nucleoid or DNA as genetic materials. Its function is to carry out genetic traits. So we're gonna see bacteria cell shape. It is divided into three major shapes, which are coccus, bacillus, and spirillum. And the coccus shape is divided again into six different shapes, which are monococcus, means one round bacteria. Diplococcus is two round bacteria, so they are together, always together. And then tetracoccus, tetra means four, so it is the shape of four round bacteria. And then sarcina is here. We can see like they make structure like cubes. And then streptococcus here is shaped like grapes. And then staphylococcus is, no, I was making a mistake. I mean, the streptococcus here is like a long chain of round bacteria, and then the staphylococcus is made a shape like grapes. It composed of many round single bacteria. This bacillus shape right here, bacillus means stick, so they have like this shape, long like stick shape, and they are divided into three shapes, which are monobacillus, diplobacillus, and streptobacillus. The monobacillus is just contained of one single bacil bacteria, and diplobacillus is contains of two bacillus bacteria, and then the streptobacillus bacteria is composed of like many single bacillus bacteria, and it makes structure like a long chain. And we have spirillum shape here, which is divided into three shapes. They are vibrio or coma, and it is the shape like coma here, and then spiral. And then we have spiroseta, which is a long spiral-like shape. We're gonna talk about archaebacteria and eubacteria differences. Archaebacteria basically has simple organization of cell, while eubacteria has more complex organization of cell. Archaebacteria can be found in extreme environments such as very hot environment, very salty environment, while eubacteria can be found everywhere on Earth. Like we said before, they are cosmopolite organisms. And then archaebacteria does not have peptidoglycan, while eubacteria consist of unique layer of peptidoglycan. And archaebacteria have introns, while eubacteria does not have introns. And archaebacteria exhibit neither glycolysis nor Krebs cycle, while eubacteria exhibit both glycolysis and Krebs cycle. And basically has three types. They are methanogens, halophils, and thermophils, while eubacteria has two types. They are gram-negative and gram-positive. And the reproduction of archaebacteria is asexual, from like binary fission, budding, and fragmentation, while reproduction of eubacteria, they are capable of producing spores. The RNA polymerase of Archaebacteria is complex subunit, while RNA polymerase of eubacteria is simple subunit. And the lipid membrane of eubacteria consists of ether-linked branches, aliphatic chains, and containing the glycerol phosphate, while eubacteria membrane lipids consist of ester-linked, and then it is straight chains of fatty acids, and they contains L-glycerol phosphate. The next part of this lesson will be guided by Miss Mery.
Okay, student, after studying general characteristics, structure and shape of bacteria, and difference between Archaebacteria and Eubacteria, now we will discuss about the reproduction of bacteria. Bacteria reproduction can occur in two stages, asexual and sexual. Asexual reproduction by binary fission, and sexual reproduction by conjugation, transduction, and transformation. Now, I will show you some videos about binary fission. Most bacteria reproduce by a process called binary fission. During binary fission, the parental cell divides to form two daughter cells. In the first step of binary fission, the bacterium replicates or copies its chromosome and attaches each identical copy to a separate location on the cell membrane. Unlike eukaryotic cells, bacteria do not need to dissolve a nuclear membrane or assemble a mitotic spindle, making binary fission very quick and efficient. Second, the cell elongates. Since the chromosomes are attached to different locations on the cell membrane, they separate from one another. Third, the bacterium pinches off at the center and forms a septum, a separating wall between the two compartments of the cell. Finally, the cell splits into two new daughter cells. These daughter cells are fully mature bacteria that can grow, and if the conditions are right, will continue the binary fission process. This is the growth phase of the Streptococcus pneumoniae, shown in time-lapse with speed 540 times. Wow! It is one cell divided into two cells, and two cells divided into four cells. And so on until it becomes 1,000 of cells. Okay, now we move to the next chapter. This chapter is the classification of bacteria. We already know that bacteria are divided into two types. There are Archaebacteria and Eubacteria. Based on their habitat, Archaebacteria are divided into methanogens, halopels, and thermoacidopels, while Eubacteria are divided into gram-positive and gram-negative. In this chapter, I want to divide you into 7 groups according to the 7 types of bacteria that I mentioned before. After that, all of groups will present the result of the group discussion. Okay, everyone understand? Let's get started. Okay students, now you all know the characteristic and role of methanogen, halophil, thermoacidophil, gram-positive, and gram-negative bacteria. And now, I will conclude what you have discussed before. First, methanogens is a archaebacteria live in an oxygen-free environment. For example, there are Methanobacterium and Methanobacillus which can produce methana in form of biogas. Second, halophils. Halophils live in areas with high salt concentration such as the Dead Sea or the Great Salt Lake. One of the halophils is Halobacterium. Third, thermoacidophils. Thermoacidophils live in hot and acid waters as in sulfur springs or deep sea thermal vents. They thrive at temperature of 70 degrees Celsius or higher. For example, there are Sulfolobus and Thermoplasma. Next, gram-positive bacteria. Gram-positive are bacteria that retain the color of crystal violet in the gram stain. For example, Staphylococcus aureus is a pathogenic bacteria that are common in humans' body. And the last is gram-negative bacteria. Gram-negative are bacteria that don't retain the color of crystal violet in the gram stain. Some of them are helpful, but some of them can cause serious diseases. One of gram-negative bacteria is E. coli, which can be found in human large intestine. Okay, that's all the explanation from me and Diah. Wassalamualaikum warahmatullahi wabarakatuh. See you.